Okay, I'll make it very short. It's an introduction to G-TwYST. It's an acronym for Genetically Modified Plants 2-Year Safety Testing, and it's the answer to a call from the European Commission on a 2-year carcinogenicity rat feeding study with the maize NK603. It's a collaborative project with rounded up 3 million euros. It has a duration of 4 years, and it started on the 21st of April 2014. And the issues related to the 2-year feeding trials that need to be addressed in a stepwise approach, that was the original text of the call, was the execution of at least one rat feeding trial with genetically modified maize NK603, applying the EFSA protocol. Participating institutions should strictly comply with all applicable international standards and norms concerning feeding trials in close collaboration with EFSA, and this is also the original text of that call. And the second point that they wanted was analyzing, reporting, and providing recommendations. In particular, as and these are two very important issues, the scientific justification and the added value of such long-term feeding trials when wanting to assess the risk of genetically modified plants. So, as I said, it's a collaborative project. We have 8 institutions which are working together in order to perform this project. And the objectives of the G-TwYST project, in answer to this call, are to elaborate a scientifically sound approach to evaluate the potential toxicity of genetically modified plants in whole feed, based on the results of extended feeding trials with a genetically modified maize NK603. And the question that Anna posed here is answered very quickly. We do it according to the OECD test guidelines, which have been published quite a number of years ago for chemicals, and have modified or taken into account many statements and recommendations by the European Food Safety Authority. And a very important issue which we are actually working on, and we will be publishing, I hope, in the first half of the next year, is the definition of criteria to evaluate the scientific quality of long-term feeding styles, because there are a number of feeding styles out there that have been published that, to make it very short, they are not worthwhile the paper on which they have been printed. To define when long-term animal feeding trials are scientifically justified and which is their added value in the frame of the GMO risk assessment process. To analyze the role or the influence of broader societal issues, including ethical aspects, normative dimensions on the ongoing debate associated with animal feeding styles in GM food feed risk assessment. The question is, we want to have a 100% safety when dealing with genetically modified plants. It's interesting to see that we don't have the discussion on the number of animals we're using in such feeding trials. It seems to be secondary or not important at all. Some are discovering it just now, and these sort of things will be discussed by Armin Spöck and his collaborators. And to make accessible the detailed scientific information, including raw data via the project website, the open access database, which has been presented and organized and established in the frame of GRACE, open access journal papers and stakeholder consultations, and to communicate the results of the project and their significance for the GMO risk assessment process to risk assessors, risk managers, a broad range of stakeholders, and the general public. So this is basically what we have planned. These are is the study design of the first 90-day feeding trial with a genetically modified maize NK603. It's basically the same study design as I showed for the 90-day feeding trials under GRACE, having 2 dose groups, 11 and 33% inclusion rates. We use the near isogenic non-genetically modified plant in a concentration of 33%, the higher dose which we use with the NK603. And as the question came only a few minutes ago, we are using the NK603 without the Roundup application and we're using the NK603 plus the Roundup application in the 2 inclusion rates I mentioned before. That makes about 172 animals which we're using alone in this feeding trial. I would like to recall that in I don't know exactly when it was, I think it was about 1 year ago or even longer, the European Food Safety Authority brought out a document in which they supported the use of maize in an inclusion rate of 50%. We had a very, very long discussion within the consortium if we should do that, and we thought this would be the opportunity to perform it. We have the big problem, the timeline, and we have started already, or we had to start the combined chronic toxicity carcinogenicity study, which I will show you in a minute, very early in this process. So that we can't perform one big 90-day feeding trial in parallel with the 11, 33, and 50%. So I acknowledge this is a problem, but we haven't been able to solve it in another way because of the animal housing facility limitation, so that we have a second feeding trial where we are going up to this 50% inclusion rate, which was recommended at that time by the European Food Safety Authority. And then the very big study, this alone takes over 700 animals. This is the combined chronic toxicity carcinogenicity feeding trial with genetically modified maize NK603. And here you see the groups again, and you will notice that the number 50 is not there. We had a very, very intense discussion at that time if we should go for a 1 to 2 year study with an inclusion rate of 50%. There was a general recommendation also, and mainly from the stakeholders, to keep the concentration below the 40% line, because if not, you would have a very big and significant protein load that would lead to nephrotoxicity in most, if not all, of the animals, and would invalidate the feeding trial. It is easy to say, well, you don't see an effect perhaps in the end because you didn't go up. I would like to know which stakeholder or expert here in the room would tell me that after 50, when we give 50% of the feed at the 2 year time and most of the animals die, who's going to take the responsibility for that? And in that discussion, we decided to stay with 33%, which I think is 1/3 of the diet being the genetically modified material. And after 2 years, you're not seeing anything, then you can't tell that I think the dosing was too low. You have here the problem, the intrinsic problem, that you can have and you will have nutritional imbalance. We will see this in the 90-day feeding trial with up to 50%. If this is a problem, there was a consensus even also with the EFSA representatives at one of the stakeholder meetings that we should go for the 90-day feeding trial with a 50% inclusion rate, and we will do so. We won't do that with the in the chronic toxicity carcinogenicity study. And as it shows here, we have 2 time points, the 12-month time point where a certain number of animals is sacrificed and analyzed, and we have the second time point after 24 months where we will sacrifice the rest of the animals, or those animals that have not had to be sacrificed before the end of the study. So this is a chart which is very similar to the one in Grace. It shows how the G-TwYST project is organized. We have a work package 1 which coordinates and manages the project, and we have then a big work package 2 which had to deal with the feed production and with the very important issue of plant analysis. We have the big work package 3 set at the Slovak Medical University doing the feeding trials in the 90-day and 2-year study. And something that is new in this project, we have definitely used a European registered toxicopathologist, have included, we are not using him, we have included a European-based and European registered toxicopathologist, Roger Allison, to perform the analysis of all the tissues which are derived from these animals according to the OECD test guideline. And then we have a big work package 4 with biostatistics. We learned already from the GRACE project that a professional group of biostatisticians is extremely important to analyze all the data we get, and Hilco van der Voort and his team at Biometris in the Netherlands has agreed to lead or to perform all the biostatistical analysis. Then the very important work package 5, where Amitsberg and his collaborators are involved on the evaluation of scientific value and societal issues of such feeding trials. We have the central access database CADIMA, so that is something we are going to very actively use or make use of. We have the very important work package 7 with the stakeholder engagement, communication and dissemination team. We have an advisory board which in all of these aspects are being put together. We have those representatives there, and we will hold the one or the other workshop with risk assessors, risk managers and other stakeholders, for example, to communicate this evaluation of scientific value and societal issues. Very shortly, this is something that I think Jochen Schiemann has already shown. It's the interaction of the GRACE and the G-TwYST projects, here showing the complementarity of the project. This is an old slide because of the stakeholder meeting which we had. This carcinogenic study with 1 dose of MON-810 was left out. This is to show how we react also to those sort of criticisms. And what is missing here, we have an interaction with other EU-funded projects, and one that is missing, I'm sorry for that, Bernard, is a very active communication and also exchange of knowledge with the GMO 90+ project. So put together, you can see how money is distributed. A very important part of the money goes to the plant analysis. This is an issue that on the first day of this meeting also became very important. About 1/3 of the cake goes to the feeding trials. We have then distributed between statistics, the evaluation, the database, the communication work package and the management package, about 25% of the money there in the project. Thank you very much for your